In this video, we will study what is translation. Translation is synthesis of proteins, and this translation process occurs with the help of amino acids. Now, amino acids, they get linked with one another to form a polypeptide chain, and this polypeptide chain forms the protein. And bond between two amino acid, this is 1, this is 2, and this bond is called as peptide bond. When many amino acids they get linked, this is called as polypeptide bond. So this formation of polypeptide, this formation of protein with the help of amino acids, this is called as translation, or we can say protein synthesis is called as translation. For the whole process of translation, what is the requirement? Ribosomes are required, amino acids are required, messenger RNA which carries the messages from DNA, transfer RNA which help in transfer of the amino acids, and an enzyme, amino acyl transfer RNA synthetase, which help in binding amino acid to the transfer RNAs. So these are the requirements for the protein synthesis. So one by one we'll discuss all the requirement. Ribosomes, they are also called as protein factories because ribosomes they help in protein formation, and ribosomes they are very specific for binding to the amino acid. So whatever information is present on the DNA, not this DNA, out of the two strands, one will act as template strand, and this will form messenger RNA. Now this messenger RNA will carry the information opposite to that of the DNA, and these are also called as codons. They are present in the form of triplet codon, and this will form amino acid. This codon will form amino acid. So whatever information is present in the DNA, that genetic information is being transferred to the messenger RNA. Now messenger RNA along with the transfer RNA and ribosomes will help in the formation of the protein. So ribosomes are also called as protein factories, like we can see here. This is the ribosome and this is the messenger RNA, and these are codons which are carrying which is helping in formation of amino acid, and this is the transfer RNA which will carry the amino acid. So next, first requirement we know this is the ribosomes, because ribosomes are also called as protein factories of cells. Now they are present in the cytoplasm, or they may be present in the ribosomal RNA. Now ribosomal RNA helps in formation of ribosomes. This help in formation of ribosomes. Now ribosomes they have two subunits. One is smaller and second is bigger. Now these two subunits they lie free in the cytoplasm, but during protein synthesis they will get associated to form the ribosomes, like we can see here. Now this is the smaller subunit of the ribosome, and this is the bigger subunit of the ribosome, and this is the messenger RNA which is binding to the smaller subunit of the ribosomes. And when these two units they are separate, they will come together only during the protein synthesis formation, and this is the protein chain which is being produced on the ribosomes during the translation process. So this is called as growing polypeptide chain. This is one amino acid, this is another amino acid. They get linked up to form the polypeptide chain. Now if we see the ribosomes, as we discussed, they have two subunits. One is smaller and second is bigger. They lie free in the cytoplasm, but during protein synthesis they will come together like this and will get associated, and for their association magnesium ions they are required. Now each ribosome has two sites. One is called as P site and second is called as A site. Now P site is called as peptidal transfer site and A site is called as amino acyl site. Here we can see this is a smaller subunit, this is the bigger subunit, this is the messenger RNA which is carrying the codons, and this is the A site which is called as amino acyl or acceptor site, and this is the P site which is called as peptidal transfer or donor site, and this is the E site. E site is formed temporary, this is transient in nature. through this site, when this transfer RNA becomes free, now this will exit. So this is called as E site. Now another thing which is present on the larger subunit of ribosome is a groove. Now this is a groove where various kinds of proteins, they can get formed like this. Polypeptide chain will be produced in this groove, and later on this will be released from this groove. So this is the polypeptide chain which is being produced in the groove present on the larger subunit. So peptidal transferase is also a component of larger subunit of the ribosome. So temporary site, E site, from where exit is taking place. So A groove is present, which is part of the larger subunit like we have discussed. So this is smaller subunit, bigger subunit, groove, P site, A site and E site. So this is how ribosomes, they help in protein synthesis. Now this smaller subunit of the ribosome, this will also get attached to the messenger RNA during the protein synthesis. Now next is, we know that next requirement is amino acid. So there are 20 amino acids which are present in the cytoplasm. Now these amino acids, if we see their structure, like here we can see here, this is the R or side chain, this is the COH group, and this is the NH3 group. So we can say on one side this will have NH3 group, positive charge. On the other side this will have COH, that is acidic group or carboxyl group. And one side this will have side chain or R1, it can be R2, it can be R3. So these amino acids, they will get linked with each other. Like if we have one amino acid, so this will get linked with another amino acid by the formation of the polypeptide chain. So there are 20 amino acids which will help in formation of the protein. So these are the amino acids which have been shown, which will get linked during the protein synthesis. Now next requirement is messenger RNA. As we discussed, DNA, this will form messenger RNA, and this messenger RNA get information from the DNA in the form of codons, which are complementary to the template strand of the DNA. Now this I have shown the structure of the messenger RNA. We can see here, this is the cap part and this is the tail part, and this is the coding region of the messenger RNA. Coding mean which is helping in formation of the amino acids, or which is carrying the genetic information from the DNA, which help in formation of the protein also. So this messenger RNA, which carries the messages from DNA, so this is called as messenger RNA, which will get binded with the smaller subunit of the ribosomes. So this is called as messenger RNA. And if you want to learn about the detailed structure of the messenger RNA, you can watch my video of messenger RNA, and link is shared in the description box. Next requirement is transfer RNA. As the name indicates, transfer RNA will help in transfer of the amino acids. They are also called as soluble RNAs. Again, if you want to see the detailed structure of transfer RNA, you can click the link which is given in the description box of the transfer RNA. Now this site is the amino acid binding site. This site is the anticodon site. So if we want to make simply a messenger RNA, we can transfer RNA we can make like this. Like this is the anticodon, which will be complementary to the messenger RNA. Now this will carry the amino acid, like supposing this is carrying one amino acid, and we have this is ribosome, smaller subunit. We know it can bind with the messenger RNA, which is having codons like this. Now this transfer RNA, this will come over here. This codon will have opposite sequence, so this is called as anticodon. Detail we will discuss in the next video. Now this is the amino acid which is binding to the transfer RNA. So transfer RNA carries the amino acid, or this help in transfer of the amino acid. So one transfer RNA can pick up only a specific amino acid, and amino acid can be specified by 2 to 6 transfer RNAs. So each transfer RNA has an area coming in contact with the ribosomes, and this also helps in binding with the enzyme amino acyl synthetase. I will discuss about amino acyl enzyme synthetase, which helps in binding of amino acid to the transfer RNA. So transfer RNA, we can see here, this is the site which is called as CCA site. This is the amino acid accepting site. And this part is anticodon loop, and this is the D loop, and this is T pseudouridine C loop. So these loops helps in binding to the ribosomes, but this carries the amino acid, and this lies opposite here, this part, which is called as anticodon loop. So this is about the structure of transfer RNA, which is helping in protein synthesis. Now this is the enzyme which is called as amino acid synthetase enzyme. This is actually the enzyme which will help in binding of amino acid to this end of the transfer RNA. So this is called as amino acid synthetase enzyme. Like we can see here, this is the enzyme which is called as amino acid synthetase enzyme. Amino acid can bind with it. Energy is required in the form of ATP. So this is first is activation of the amino acid. This will discuss how amino acid, when they are lying in the cytoplasm, they are passive and they have to be activated with the help of amino acid. So amino acid with the help of ATP will get activated. When this gets energy, this gets activated. Like ATP has 3 phosphates. So out of the 3 phosphates, one will bind with the amino acid and amino acid will get activated. Like we can see here, this is amino acid, this is ATP, and this is the enzyme to which this is binding. So this get activated and pyrophosphate is released. So later on, this amino acid will bind to the transfer RNA, and transfer RNA, when this binds, this is occurring with the help of enzyme amino acid synthetase enzyme. At the end of the reaction, this enzyme will become free. So function of amino acid synthetase enzyme is binding of the amino acid to the transfer RNA. So this is the transfer RNA which is carrying this anticodon, and this site is for binding to the amino acid, which is being done with the help of amino acid synthetase enzyme. So we have seen in the whole video what is the requirement for protein synthesis. We need to have ribosomes, which are also called as protein factories. Like this is the ribosomes which get associated during the protein synthesis. Then we require messenger RNA, which is carrying the codons or in genetic information from the DNA. Then we require the transfer RNA, which is carrying the anticodon part, and this is also having this is linked to the amino acid. So this is the amino acid. So these messenger RNA, transfer RNA, then this ribosomes and enzyme amino acid synthetase enzyme, which help in binding of amino acid to the transfer RNA, and transfer RNA carries the amino acid over the ribosomes and messenger RNA for the formation of the protein. So these are all requirements for the protein synthesis. In the next video, we'll discuss about the exact mechanism, how protein synthesis occurs. Thank you for watching my video. If you like my video, please like, share and subscribe. And you can also ask me questions, and you can share those questions in the comment box. Thank you for watching.